And I will be also the TA for a couple of first labs, I think, physiology labs. We'll be dealing with neurophysiology for about 8 weeks, and you will learn the details, what is the subject matter, especially in labs, during the labs, and what kind of tests will be applied to check your knowledge. Right now, I have the first lecture from neurophysiology, introductory neurophysiology, as I said previously. The next couple of lectures will be provided to you by my colleagues. Then I will have again the lecture, I think, on higher activity of nervous system. You can find it all. It will be visible in front of the Department of Physiology. There will be information on that, and you will have the subject of the lectures provided to you. If you cannot find it, you can come to me during the labs, and I will tell you in more detail what are the subjects in the labs and who will be the TA for given labs. Today we are starting with basic neurophysiology, thinking about how the nervous system in general works, what are the rules as far as function of central nervous system goes, and also what are the biological basis for function of nervous system. So very basic information based on the high school material, basically. But I would like you to prepare yourselves for the labs, because I expect that you will be able to use the knowledge which will be shown to you during the lecture. And please do recall some information from high school so we can use this knowledge for completing the first lab for English students. Okay, so when we are talking about nervous system in general, the first question it's logical to ask is, what is the general function of this system? And we can name several functions we associate with nervous system, but on the very general level, we can say that nervous system, the main function of nervous system, the distinguishing function of nervous system, is conducting and analyzing, sometimes generating information. So it will register the changes in environment, conduct this information to the central nervous system or lower levels of nervous system, and then analyze it and send the signals in reaction or in response to those environmental changes. Sometimes nervous system will generate spontaneously the information and send it to different parts of the body. For instance, the system which is directing our breathing will be rhythmically producing the bursts of information to control the breathing rhythm. The central nervous system pacemaker will spontaneously regulate our internal circadian rhythms. So these are the examples of generation of information within the nervous system. So it's not only passively conducting the information, but also there is a lot of analysis of information and also the generation of information. So it is reasonable to ask, what is the information? How we understand the information when we want to understand how the nervous system works. And I will give you a very simple definition, but the definition which is quite consequential for our understanding of biological mechanisms associated with information conduction and analysis. In every known example, either information in organic system or living systems, or information in inorganic systems, information is always associated with the certain disturbance in the environment it is conducted. So the information cannot be generated or cannot be conducted, cannot be spread in a vacuum. There always needs to be some kind of environment. It can be the wire where you can pass the current, or it can be even the bubble of air where you can change the pressure of the air within this bubble and it can contain information. Or it can be even the surface of water where you can disturb it and you will see the waves spreading around, and this can encode the information as well. So in general, the information or appearance or presence of information is associated with the disturbance in the environment. It may be very different environment, various environments where information can be spread, but it needs to be material or material environment. Without that, you cannot spread the information or you cannot conduct the information. So that said, let's think what are the consequences of this nature of information. What does it mean that this disturbance in the material environment? There are two important consequences of this definition or this fact. As a matter of fact, it is true. This is what is material appearance of information is. Okay, so there are two basic consequences of this, and both of them are quite important for the function of nervous system. First is that because this is disturbance in the specific disturbance in the environment, it is important in order to be able to send the information and receive the information, then that environment during this process of sending or conducting the information is in the resting state. So we can distinguish disturbance, which would be called or which would contain encoded information. When you have a lot of noise in the environment, then you will not be able to detect the change which contains information you or system is interested in. So say if we would be in the real room and I would be lecturing to you or anybody would be speaking, it is preferable that only this person is speaking and not everybody is talking about whatever, because then the noise in the environment, in this case, voice environment or the air around us is so high that we may not be able to extract the information which is destined for you, meaning my voice carrying certain message. So this is the first condition for information conducting system to work. This first condition is the environment in which the information is spread must be in the resting state. The second consequence is strongly associated with the spreading or conductance of the, or spreading maybe is a better word, spreading of the information. Because information is the disturbance or a disturbance or given disturbance in concrete environment, when it spreads in this environment, move certain distance, the environment is resistant to this spreading. The energy is dispersed in this environment. Resistance is the feature basically of all material environments. So when the disturbance is spreading, it will be changing due to the resistance exerted by the environment itself. When you think about say you drop the stone in the lake which surface is in resting states, so there are no waves in this lake. When you drop the stone, you will see that waves from the point of droppage are spreading in the circular manner. But the farther they are from the point of droppage, the lower the amplitude of these waves. So this disturbance, which potentially carries some information, is losing amplitude, is losing energy, and as consequence, the amplitude of the waves is smaller and smaller, the farther the information or this disturbance travels. There is a specific term used in neurophysiology for this phenomenon, and this loss of amplitude is called decrement. And when you think about it, it is a problem, because in nervous system, information sometimes is conducted for long distances in axons. As you can see on this slide, which we have right now, the axons are the parts of the nervous cell or neuron which are responsible for sending information far away. And this system, which is involved in spreading the information over long distances, must take into account in its mechanisms the fact that there is decrement when the given disturbance in the environment of the neuron or axon in this case is spread over long distances. One can expect the decrement, so the amplitude of this information will be smaller and smaller with the distance. As you will learn, this phenomenon of losing the amplitude or phenomenon of decrement was taken care by biological system, and you will learn how the systems dealt with that. So the systems are maintaining, nervous systems are maintaining the ability to send the information and code it in the change of environment over the long distances. Okay, so these are the two basic properties of the information spreading. Let me remind you. First one is associated with the fact, or both of them are associated with the fact that information is some kind of disturbance in the environment, therefore, or has a form of disturbance in the environment. Therefore, in order to conduct or transfer the information effectively, you need the environment in the resting state in order to be able to spread this information, firstly. And secondly, the fact that this is material environment in which this disturbance is spreading or is traveling causes that the energy of this change, of this disturbance, which encodes the information, is being lost over the long distances. So amplitude is decreasing. So let's now go into the biological substrates or biological elements of neuronal structures, which are responsible for conducting the information in the nervous system. Before we go there, let's first think for a moment about typical neuron. Especially, I would like you to look at this slide that I have up here. And let's look at the different parts of the neuron. Each, or not each, because it's different for different types of cells, but the typical schematic structure of the neuron is that it has the so-called dendrites. Then there is a perikaryon, basically cell body, and then axonal filament or axon. I named those parts in the order which reflects how the information is traveling in the neuron. So usually the dendrites act as or have a function of some kind of antenna for the neuron, and the numerous dendrites are collecting, you can see them in the upper part of this slide, numerous dendrites surrounding the cell, and they are acting as antenna, collecting the information from environment. There are information encoded from the environment, for instance, from some kind of synapse, the information from the synapses, but not only synapses, generally from environment, is detected by dendrites, and this information is encoded in ways which we will discuss later. And this information is sent through the cellular body, so it goes to the cell body. It's described on this slide, so you can see. And then it goes to axon. And axon, as you can see on this illustration or on this drawing, is only singular. There's only one place in the cell body where it starts, and usually it may have some collaterals. So it will distribute the information to different surroundings, but it's basically one filament. It starts in one place, and only one collective information is sent through the axon. So when we look at those morphological features of the neuron, we may associate them with the function. We know now that dendrites are responsible for collecting the information. We know that axon, singular axon, is responsible for conducting or spreading this information, usually to far away places far away from the cell body. And this, the structural elements of this organization of the neuronal structure suggest what is the function of cell body. Look at this that way. There is a lot of dendrites sending the information into the cell, but only one axon which sends the information from the cell. It means that the cell integrates in the cell body, integrates all the information from dendrites, and then sends it farther via axon as a synthetic single message. So you have a lot of messages coming on. They get analyzed in cell body. In the classical neurophysiology, it was understood this way, and it's still true in many systems or in many neurons. And then it gets to be sent via axon towards other cells or different levels of nervous system. So when we are talking about encoding the information and certain requirements which are associated with that, with the spreading of the information, the most distinctive feature, for instance, of the axon is the feature that it sends the messages or information over the very long distances, or it may send the information over very long distances. So this problem with decrement in the passage of the information I mentioned to you is especially visible in this part of the system, because dendrites and cell body in this part, excuse me, in typical cell, the information is traveling over relatively short distances. So we'll look in this very basic preparation for understanding the system. We'll look what are the substrates for this information passage and different types of information passage in different parts of cell body. So let's first look at the structure, which you should be, excuse me, quite familiar with, which is crucial for transfer of information encoded in the electric changes. And this structure, as you, I guess, know, is cellular membrane. Cellular membrane is quite complex structure, here is the better maybe picture of it, quite complicated, both structurally and functionally structure, but we can distinguish or indicate two basic functions of this structure of the cellular membrane. And they seem to be, in a sense, opposite, but there's, of course, justification for that. Firstly, cellular membrane task is to help or enable cellular internal environment to maintain its integrity. For living cell, it is important that it maintains specific for given cell content of the inside of the cell. It's radically different than the environment surrounding the cell. So in order to maintain this internal environment, specific internal environment, cell has to be bordered in the sense, and the cellular membrane plays this role of isolating the internal environment from external influences. The second function, which seems to be also opposite to this one for cellular membrane, is a communication function. Cellular membrane has the structures which are necessary for the communication between inside of the cell and the outside of the cell. So the structures are associated, for instance, receptors are associated with sending the information from the environment into the cell and vice versa, so from the inside of the cell to the environment. These two opposite in a sense, but they are playing in the concert in a sense. These two opposite functions are associated with specific structures within the cellular membrane, and those structures are built by specific substances or specific molecules. So the isolation part of the cellular membrane function is associated mainly with the phospholipid bilayer. This phospholipid bilayer, here you can see this red heads and yellow tails of fatty acids. These are the elements of phospholipid bilayer. So this part, it will elaborate a little bit more on that. This part of the cellular membrane is associated or is responsible for isolating intracellular environment from extracellular environment. The structures which are, or molecules which are strongly associated with the communication function of the cellular membrane are proteins, especially these integral proteins. This blue one, you see the internal protein, it's described on the slide. And the characteristic feature of this protein is that it penetrates across the membrane. So you have part that is in touch with extracellular fluid and part that is in touch with cytoplasm. So by their structure, those proteins are predestined for communication function between internal and external environment because they participate somehow or are immersed in both of those environments. So let's have a look at the bilayer first and think why it can perform this isolating function. As you are aware, I guess, the phospholipid bilayer, as the name indicates, consists of phospholipids, which have so-called phosphate head, which is hydrophilic, and the fatty acid tails, which are hydrophobic. These properties of every phospholipid molecule causes them, if you have many of those phospholipids in the solute, like water or solute containing water, they tend to spontaneously create the default structures, which are either bilayers or micelles These are small bubbles, or a little more complicated structures called liposomes. And the mechanism of that is that the tails, fatty acid tails, are turning onto themselves when you look at bilayer structure, because they are hydrophobic. And hydrophilic phosphate heads are turning towards the water. So this group of molecules spontaneously creates this kind of structure. So you don't need really living cell to get this kind of structure. And there are certain characteristic features, functional features of this bilayer, phospholipid bilayer. The one that is directly associated with the function it is responsible for, meaning the isolating function. This feature is can be described as the high level of or low level of permeability of this phospholipid bilayer, meaning that very few molecules can freely cross this bilayer, phospholipid bilayer. And the only molecules that are characterized by small size, it's 2 or 1 atoms only. So this is the first condition for the molecule to pass easily the phospholipid bilayer, small size. And second feature, which is very important for us, is that those molecules which are able to cross the bilayer don't have electrical charge, meaning that no matter how small the molecule, if it has electrical charge, it will not pass easily through or at all through phospholipid bilayer. Putting it into different words, the phospholipid bilayer is very good, very efficient electrical isolator. It isolates the internal part of the cell electrically. I'm sorry, there's some technical issue, but I hope that we can go on now. Okay. So it is a good electrical isolator, and it keeps also larger molecules out of the, or does not allow simple crossing of larger molecules. This is the basis, this property of this bilayer is the basis for its isolative function in the cell or for the cell. When you look at this little insert, black and white insert on the left side of this slide, you will see the schematics of the phospholipids in phospholipid bilayer, and you will see these arrows are showing you that those phospholipids are not static. One of the features, important features of the phospholipid bilayer is the feature or the fact that this structure is dynamic. It's not stiff. The phospholipids are moving there all the time. And as you can see, there are different types of movement. There you have like spinning around, which is marked by A. B is like shivering, and C, you have longer distance migration associated with the movement of these phospholipids within the layer. Sometimes it's not very frequent, but it was observed those phospholipids can flip it's called flip-flop. They can flip from one part of the layer to the other, so say from upper to the lower, as you can see in this D example on this insert. So there is constant movement of those molecules. It is important because this is the environment in which our proteins are sitting. And because the structure is dynamic, the phospholipids are moving all the time. This causes the minuscule but functionally noticeable and important changes in structure of the proteins which are sitting in this dynamic bilayer. We'll come back to that certain point, and you will learn that it's quite important, this changes in the protein structure, especially the tertiary structures. It's important for functioning of the whole system of information transfer. Right now, just remember that this is dynamic structure, and that because the phospholipids are moving around. Before we move on, let's ask the question first, why they are moving. And of course, in order for any molecule to move, you need kinetic energy. And this kinetic energy is derived for thermal energy or heat energy. This energy in the universe, not only in our organism, but in the whole universe, is relatively easily available, because this movement of the molecules will be present, of course, to different degrees, but will be present always when temperature is higher than absolute zero. So when you have the temperature at the level of absolute zero or below, which cannot be obtained in the laboratory setting, we cannot get this kind of low temperatures, but the physical models indicate that the molecules will not move in this situation. But because since the Big Bang, this initial explosion that created the whole, or was the beginning of the universe as we understand it, since that moment, the temperature across the whole universe is higher than absolute zero. So basically, molecules are moving. Keep that in mind, because this is not only important for this bilayer, but it will be also important when we discuss the processes of diffusion. So also, you need to know and you need to remember that the term for this motility of phospholipid bilayer is called sometimes fluidity of the membrane. So the more of the motion in phospholipids, the more fluid the membrane is. And when you are doing research on that, you can observe it directly. The method which is used is I think, pretty simple and pretty illustrative of the process. You just create one phospholipid which is emitting fluorescent light. You put it into this given bilayer, and you observe how fast it moves. If it moves slowly, it means that membrane fluidity or overall movement within the membrane is relatively small. When it moves fast, the membrane fluidity is high, and the overall of movement in molecules in this membrane is relatively high. As I said, the membrane fluidity is important because it will determine the certain features of proteins which sit within the membrane. So because the kinetic energy which is used for the movement of these molecules is directly derived from this thermal energy associated with Big Bang, very, very far away in terms of time from now. So it's somehow obvious in the logical way that the factor which will be influencing the membrane fluidity, important factor, is temperature, environmental temperature. So if you have membrane which is placed in the environment with changing temperature, the level of fluidity will change as well. The higher the temperature, the more energy, the more movement of the molecules, and the higher fluidity. So higher temperature, higher fluidity. Indirectly, you can guess the importance of the temperature and the stable membrane fluidity for the development of nervous system in general, in the evolutionary sense. This observation is that only the animals whose nervous systems are developing in relatively stable temperature are capable of developing complicated nervous systems, complex nervous structures. And of course, the group of organisms which will be able to keep the temperature of their internal temperature at a relatively stable level are the organisms which are capable of producing enough metabolic heat inside their bodies, so-called endothermic organisms. They have relatively stable internal temperature, no matter what, or independent from large influences of the environment, external environment temperature, or this complex, and these are mammals, birds, so these are the groups of animals which are capable or developed in the process of evolution most complicated nervous systems. And one of the factors which allowed for that is maintaining the stable internal temperature. You can also observe the relatively complex structures of the nervous system in organisms which are living in a stable thermal environment. So despite the fact that they are not developing independence from environment as far as temperature maintenance is concerned, their internal temperature is still relatively stable because their environment is stable. So when you look at the octopus and its quite impressively developed nervous system, this is associated with the fact that the environment of the ocean, the areas where the octopusi are living, has relatively stable levels of temperature. So the membrane temperature and therefore membrane fluidity is not changing that much. And this allows for precise communication, but you will learn how it works a little bit later in the course of neurophysiology. Okay, so this is this one factor influencing the membrane fluidity. I want to point out to you that there are other factors which will influence the level of membrane fluidity, also in humans, in us. So one of the chemicals or one of the molecules that would influence how movable phospholipids are within the bilayer, so how much fluidity there is the molecule of cholesterol. Cholesterol tends to bind to phospholipid molecules and therefore limits their movements. So the higher level of cholesterol present in the membrane will decrease the membrane fluidity. There will be less movement of the membrane, which is not that good actually. But still you need some cholesterol there for the proper structural properties of the bilayer. Also the molecule which influences strongly membrane fluidity is the phospholipid and structure of the phospholipids, and in particular molecular structure of the fatty acid tails. So as you can see here, the fatty acid tails consist of carbon atoms with attached hydrogen atoms. And the bonds between carbon atoms, you can see in the upper left corner of this slide, can be connecting via singular or double bonds. And we call, when this connection is double bonded, we call this connection that it is unsaturated, because it's not fully saturated with hydrogen atoms. Okay, if the bond between carbons is fully saturated, there is only one bond. But when it is unsaturated, there can be double bond. And when you have single bond, look at the lower right panel. So you have the saturated bonds only between carbon atoms. The structure of the phospholipid is pretty symmetrical, simple, and relatively elastic. So when it even moves, it does not introduce that much disturbance in the environment of the membrane. Look at this model of phospholipids in the upper part of the slide. You can see that one of the chains, fatty acid chains or fatty acid tails, is saturated and it is relatively straight in shape. The second one, the left one, is unsaturated. They call it here monounsaturated, meaning that only one bond is double, the rest is single, but it already introduces certain asymmetry in the molecular structure, and this asymmetry is relatively stiff, so it doesn't easily bend, in other words. So when this kind of molecule is moving within the membrane, it causes much more disturbance than saturated parts of the molecule. It's due to the fact that its reach is much larger and it is not flexible. It's relatively stiff. You can compare it to the fat and thin person entering the crowded bus. When the thin person gets into the crowded bus, it can squeeze through the crowd of people with relatively low disturbance. But if someone is really big, it causes a lot of more disturbance in this group of people. So this is somewhat, I thought, similar to the effect of unsaturated bonds on the membrane fluidity. Also, it's worth mentioning that one of the factors which will change your membrane fluidity is ethanol. It increases the movement within the membrane, it increases membrane fluidity, and actually some effects of the increased exposure of nervous system to ethanol are associated with changes in membrane fluidity. So it's worth keeping in mind, and be careful with ethanol, basically. And the fact that also there is certain, when you observe somebody who's drinking a lot of ethanol over a relatively short time, of course you will observe the slurred speech, but also you will observe that the first of the system, which is first parts of the nervous system that are affected are the parts responsible for controlling posture and movement. And these are particularly sensitive to these changes in membrane fluidity because there is a lot of complicated processing of information going on there in order to keep our posture and our movement correct or smooth. So when you change in physiologically not controlled or not controlled in a physiological manner level of membrane fluidity, you will see those effects. It is believed that some anesthetic substances, especially the ones which are inhaled, are also acting by modifying membrane fluidity. Overall, quite important parameter. Let's move on now from the isolating function to the function of communication. And before we do that, we'll have the 5-minute break, then we'll go back to the subject matter. So it's now right now 3:57 p.m. Let's meet at 4:02 p.m. I will be back and we will continue. Hello, I'm back. I hope you are back as well. So we discussed the structure of the phospholipid bilayer and its involvement in the biological membranes in isolating the internal environment of the cell from the external environment. So now let's look at the structures which are responsible for communication between inside and outside of the cell. So those structures, as you probably are aware of, are proteins, as I mentioned, especially integral proteins. And one of such proteins, well, let me come back for a moment. Just let me enforce the observation, which I think I mentioned before the break, that bilayer is a strong, very good electrical isolator. This is very important for our understanding of conducting of the information within nervous system. So please do remember that it has bilayer, phospholipid bilayer, has in general isolating properties. Especially importantly for us is the fact that it is electrical isolator. Let's look now at the proteins which are contained within the structure of cellular membrane and embodied in this dynamic phospholipid bilayer, and look at their communicative properties. And because for the processes we were talking about, the electrical currents generated by ions across the membrane, neuronal membrane, are particularly important, we'll look into the proteins which are involved in transportation of ions across the membrane. The probably very well known for you protein or group of proteins that are involved in this kind of ionic transport across the membrane are ionic pumps. These are structures. You can see this, the illustration of the how they work on this slide. So these are structures that are basically moving the ions by their nature. Ions are carrying electrical charge, so they cannot pass the bilayer, phospholipid bilayer easily. They cannot actually pass it at all. So you need the transporters for those molecules. And as I said, one of type of transporters are pumps, ionic pumps, and one which is very numerous in basically all cellular membranes is sodium-potassium pumps. It's very important. It's very dense, especially in neuronal membranes, and it uses a lot of energy because it transports the ions across the membrane actively, meaning that it transports the ions with the use of biological energy contained in ATP in this case. So when during one turn of this single pump, sodium-potassium pump molecule, as you can see it here, the 3 ions of sodium are moved towards outside of the cell. So they are moved from the cell to the extracellular environment, 3 sodium ions. And at the same time, you can see it on both actually parts of the illustration here. So 2 potassium ions are moved into this cell. And this is all this pump is doing. Basically moving these ions to the outside and to the inside. Sodium out, 3 ions, potassium in, 2 ions. This in some neuronal cells, it takes up to 2/3 of all energy that is used by the cell. So if you have, say, 2 or 3 sausages in the morning for breakfast, energy from 2 of those sausages will go to the transport of sodium and potassium ions in some neurons. Quite a lot of energy. So this fact that this structure is very conservative from evolutionary standpoint and is so highly energy consuming indicates that the function of the structure is important. And indeed, it is so. We will learn how important it is in the course of our discussion today and the other days. But let's think for a moment, what are the consequences of such action of the sodium-potassium pumps? What happens? What are the effects of this mechanism or modus operandi of those pumps? So you can easily deduce from this simple picture or the simple knowledge what is happening. Firstly, we would observe that during the process of moving these ions, 3 positive charges are moved out of the cell, and only 2 positive charges are moved into the cell. That means that due to the activity of those pumps, which are very numerous, densely packed in the cellular membrane, the negative charge is growing inside of the cell. This work of the pump is causing a deficit of positive charges. Therefore, the net charge within the cell is becoming or is negative. So this is so-called electrogenic effect of sodium-potassium pump work. And in this electrogenic effect, it's not that important what are the carriers of the charge. The chemical nature of the carriers of the charges is not that important. The important fact is that these charges are moved in certain proportion, meaning that net effect is that with every turn of this pump, 1 positive charge is removed from the cell. Hence, increase in negativity of the intracellular charge, because there are 3 positive charges associated with sodium and out, and only 2 positive charges associated with potassium in. So electrogenic effect of the pump work. The second effect, which is also of importance for us to understand how the system works, is the chemical effect. And in this analysis, or in this part of analyzing the effects of pump working, we'll focus on the chemical nature of the charge, of the carriers of the charge. So as I mentioned, this is sodium and potassium. So because 3 ions or 3 sodium atoms, it's not important in this chemical part that they are ions. The important part is that these are sodium atoms are moved out, and 2 potassium ions are moved or atoms are moved in. Of course, we easily can see that the action of the pump is causing decrease of the density or concentration of sodium ions inside of the cell compared to the outside of the cell. As a result, the concentration of sodium ions outside of the cell is much higher than concentration of those ions inside of the cell. Inversely, or opposite to that, the potassium ions are transported into the cell every time there is a turn of the pump. So the level of potassium inside of the cell is increased, and it is concentration of potassium inside of the cell is larger or higher than outside of the cell or in extracellular fluid. These are the two very important consequences of the pump's work. There is also the pumps, especially sodium potassium pumps, are also important for maintaining the water balance of the cell. As you know, the water molecules can be by layer. It's not very easy for them, but they can do it without any carriers. So because the concentration of all substances within the cell is high, much higher than outside of the cell in extracellular fluid, so the water is in a sense leaking all the time into the cell. So this has to be taken care of, and this is done by these sodium potassium pumps as well, because each ion, when it is transported with so-called water coat around it. And sodium has the larger coat than potassium. So there's more molecules of water associated with sodium ion than with potassium ion. So as you can see, as you can expect, the ions, sodium ions, are pushing or carrying a lot of water molecules with them, much more than potassium ions that are only two, first of all, and secondly, they have a smaller coat, water coat around them. So actually, pumps are not only regulating ionic balances, not only charge and chemical character of the inside of the cell, but also they have this huge responsibility, because otherwise the cells will be bursting. They are removing excess water from the cell, inside of the cell. So this is their additional overall important function, but this is not directly associated with passage or spreading of the information, the ways the information is conducted within the neuron. When you look at the schematics, which came on right now, you have basically the summary of what is happening due to the activity of the pumps. And you can see this electrogenic effect of the pump. Excuse me for a moment. You can see this minus in the brackets in this schematic cell on the upper left corner. This indicates that the inside of the cell has negative charge when compared to external environment. The result of these differences of the charges between inside and outside of the cell is that on the cell membrane you can measure certain potential, electrical potential, in model of the cell neuron. It's all usually set at minus 70 millivolts. When you have potential on the membrane of the value which is minus 70 millivolts, it means that there is more negative charges inside of the cell than outside of the cell. So this is electrogenic effect caused by the pumps, and the rest, what is illustrated here, indicates the effects of pumps and the metabolic machinery within the cell. So you have big symbol of potassium ions, because there is hydrogen inside of the cell. The symbol is inside of the cell. Then you have outside of the cell large sodium representation. There is a higher concentration of sodium outside of the cell. And you have also the chloride ion, which is in higher concentration outside of the cell. You can also see this negative ion. These are organic anions. So negative ions, which are associated with large organic molecules like proteins, sugars, their net charge in the cell is negative. So they contribute to this overall negative charge within the cell and contribute to the membrane potential, which typically or schematically is around minus 70 millivolt in typical neuron. On the schematics in the upper left corner, you don't see one important ion, which is calcium ion. It is placed in the table down here, and this is important. It plays a lot of important functions. And as you can see, outside concentration, extracellular concentration of this ion is much, much larger than cytoplasm concentration of this ion. This is worth remembering, along with the concentration gradient for potassium and sodium. These are things to remember. Excuse me. Okay, so this is the effect of the pump activity. In general, pumps and importantly, sodium-potassium pump are performing so-called housekeeping functions. They are maintaining or responsible for relatively stable internal environment of the cell in respect to ions and electrical charge. So because they are playing this housekeeping, so-called housekeeping function, they are always active. There is a lot of activity going on in the cell on the surface of the cell. So this housekeeping activity must be going all the time to keep the internal environment relatively stable. As you can guess, it is associated with necessity of stable resting environment when you want to send an information in this environment. So these pumps are, we are saying that they are working constitutively all the time. They don't need a particular stimulus to start working. They work all the time. They transport, let me remind you, transport the ions via the process of active transport with the use of biological energy. And what is also important, and I didn't mention it yet, their transport of the ions is selective. So the sodium-potassium pump is transporting only sodium and potassium. The sodium-calcium pump will transport only sodium and calcium, and so on. So these are the three basic features of the pump activity as one of the proteins responsible for communication between inside environment and outside environment. This activity is associated or is resulting in the phenomenon which is called resting potential. Because the pumps are responsible in a major way for a difference in the charges between inside and outside of the cell, the resting potential in large part is produced by those pumps. And resting potential is the membrane potential which is maintained on the cellular membrane of the neurons, but not only neurons, other cells as well, during the time when there is no transfer or generation of information in this cell. And here in this table, you have three factors which are important for generating or origin of the resting potential and maintenance of this resting potential at the certain level, namely in this model cell at the level of minus 70 millivolt. So you have here large intracellular organic ions. We talked about them moments ago. They are sitting inside of the cell, they are large, and they contribute to this negative resting potential. Third point in this table is electrogenic result of sodium-potassium pump activity. There is a lot of orthographical and grammatical mistakes in this point 3, which makes it particularly interesting from the semantic point of view, but it should read electrogenic result of sodium potassium pump activity. This would be correct formulation of this point. Anyhow, so please excuse the mess here in this third point. We did talk about that. It should be pretty clear how it happens. And finally, point 2 will become clear in some time to you, maybe even at the end of today's lecture. But it's a very important factor is ion concentration gradients across the cellular membrane, in particular potassium gradient. And this gradient, of course, is the result of long-term activity of sodium-potassium pump, and it is very important for stabilizing the membrane potential, resting membrane potential. At the end of today's meeting, I will give you the tools to think about it, and please be prepared that we will be discussing it. I will be asking you a couple of questions during the lab about how come the potassium gradient is important for maintaining the stable resting potential on the cellular membrane in general and in neurons in particular. Okay, the second class of proteins that are associated with the transportation of ions across the membrane are ionic channels or ion channels. And the very important difference between the ion channels and the ionic pumps is that firstly, transport through those channels of ions is passive. This is passive transportation. And second important difference is that they are not, in most cases, majority of cases, they are not transporting the ions in a constitutive manner, so not all the time. They are transport or allow for transportation of those ions only under the influence of environmental changes. So they basically, those proteins, react with structural changes to changes in environment. Basically, they are reacting to certain stimuli which appear or show up in the environment around the cell. Due to this fact that they are reacting to the external environment changes, or sometimes even internal environment changes, we can systematize those proteins, these ionic channels, according to the type of stimuli they are responding to. I mean, so we have voltage-gated channels, mechanical channels, ligand-gated channels, and very broad group of others, which are not clearly defined. Okay, voltage-gated channels, they will be reacting to the changes in the membrane potential. When the membrane potential changes, they will change their structure and usually open or close. Mechanical channels are the structural proteins that are strongly chemically bond to the membrane. So the displacement of the membrane, bending of the membrane will cause the change in their structure. They will be opening, closing, and so on, under this kind of influences. Ligand-gated channels contain in their structure allosteric proteins. And allosteric proteins are the proteins that will change their structure when the ligand binds to them. So, for instance, the channels which will change their structure and state from closed to open that are reacting to neurotransmitters are this type of channels, ligand-gated channels. When the neurotransmitter appears in the synaptic cleft, excuse me, the molecules of neurotransmitter will bind to the channel structure and cause the change in its structure. And finally, this capacitive others group, there are channels which we don't quite understand how they work. And this group of channels, in this group of channels, we can include temperature-dependent channels because we don't quite understand how the change in temperature causes the change in the structure of the channels. And the leak channels. Leak channels are very important. They were discovered relatively late in the science dealing with channels. And they are different from other channels in this, that they stay open most of the time. So in a sense, they behave just a little bit like pumps, because the transportation of ions can happen there all the time. Of course, all the channels are different from pumps in this is passive transport. In pumps, we had active transport. In channels, we have passive transport, meaning that stored energy in biological organic molecules is not used for this transport. Okay? By the way, just homework for you, think, because if you have movement, if you have transport, you need energy, even if it is so-called passive transport. In active transport, you have energy from organic molecules like ATP. In passive transport, the source of energy is different. I'll think for a moment, what is the source of energy for this passive through the channel transportation of ions? Anyhow, so they are different in this important two ways. Firstly, they don't usually transport ions constitutively. They are reacting to the changes in the environment, changing their structure and opening and letting the ions through. And the second important difference is that this is the passive transportation, not active transportation. So this presence of those structures in the membrane changes, in a sense, the situation of the whole system, because so far we dealt with a relatively stable, not dynamic system, which consists of phospholipid bilayer and pumps, which maintain relatively stable internal environment of the cells. And now we added channels which will dynamically react to the changes in the environment. So we would have to ask ourselves the question, okay, if, say, sodium channels will open in this resting situation of the cell, when you have resting potential of the membrane, what will happen when you cause the membrane by opening the sodium channel to be permeable to sodium? And we can answer this question, but first we have to think what forces will influence the movement of ions across the membrane when it is, the membrane, permeable to those ions. Because the ions are have somehow double nature. On one hand, they are molecules, but also they are charges. They are molecules with attached charges. So when we are discussing the forces that are acting on those structures, double character of the structures, we have to take into account the forces associated with the fact that these are chemical molecules on one hand, but also we have to take into account forces that are associated with interaction between charges. Okay, so the first group of forces, so-called chemical forces, which are universal and acting on molecules, are the forces of chemical diffusion described by Fick's law. And there are two basic rules which will be useful for us when we try to analyze the movement of ions across the membrane. Sorry. Okay, so first law, important for us, is that when you have two environments with solutions of given molecule, and they are separated by permeable membrane that is permeable to those molecules. And there is a difference in concentration of the molecules in those two compartments. So we say that there is a gradient of the concentration of given molecule. The molecules from the higher concentration environment will travel to the environment of lower concentration. This process is called diffusion, and the force which causes this is called chemical force. And first rule here is that the movement of the vector of the force is directed towards the environment of lower concentration of the molecule. The second rule about which we should remember is the rule that says, it's the second Fick's law, that says that the strength of this chemical force depends on how large the gradient is between the two environments we are analyzing. So the higher difference in the concentration between these two environments of given molecule, the stronger the chemical force that is generated and is pushing those ions through net flow, causing net flow of those ions through the permeable membrane. So when you are thinking about ions, they are molecules, therefore you have to think about chemical force. But it's not the only force you have to take into account. The second force is electrostatic force, and you have to take into account this force because these are not only molecules, these are ions, so they are molecules with electrical charge. And these electrostatic forces are governed by Coulomb's law, which described electrostatic interactions. And again, two rules which are important for us. First is the rule that says that the same sign charges are pushing each other apart. And if you have two different signs of charges, so minus and plus, these two different charges will pull each other or will pull each other towards themselves. So you have push away and pull together. When they are different signs of the charge, they will pull themselves together, and when they are the same, they will push themselves apart. So this is important to remember. And the second conclusion from the Coulomb's laws is that the size of those electrostatic force is proportional. You can see it in the equation here in the lower right corner. The size of this force or length of the vector of the electrostatic force is proportional to the size of the charge. The larger the charge, the bigger the force. And these two sets of laws will help you to analyze the possible movement of the charges across the membrane. Not charges, sorry, ions, because for charges you can use only Coulomb's laws, and you have here molecules and charges. So I think I will stop on this presentation, this lecture, but I would like you to think a little bit about it before labs, before you come to the labs in the second half of the week, and think about those forces I discussed with you at the end of this lecture in terms of how they will influence the movement of the ions across the membrane when the channels for given ion will open. So first you have stable resting state when the membrane is not letting any ions through. Then the sodium, say, channels open. So the membrane becomes permeable to sodium ions. And they can flow relatively freely through this membrane using those two sets of laws, Fick's laws and Coulomb's laws. Try to figure out what will be the direction of movement, either of sodium ions or potassium ions, for instance, when the membrane becomes permeable to those ions. So it's coming out of the resting state. All right, so I think for today it's enough of basic knowledge for you. I hope technically it all worked. I will see you in the second half of the week during the labs. If you have any questions to this lecture, try to think about them and I will be happy to answer them during the labs. If you have some very urgent question to me concerning this part of the knowledge I'm passing on to you or reminding you of, you can use, there is some mechanism here. You can like say that you want to ask the question and I should see it on my screen. But for the lecture itself, we are done for today. Thank you very much. Bye-bye.
Thank you, Professor. Bye-bye.
Thank you.
Thank you. See you.
Thank you.
Thank you.
Thank you.
Thank you.
Thanks.